So what I'm going to talk to you about is the specific application of a lot of these tools that are now in development. And it's not just the tools of biology that Eric talked about, but it's also the tools of getting through the regulatory process that David talked about. These are all things that we need to take into consideration. I'm not an expert in this, but it's amazing how fast you can become an expert in something when it's necessary in order to move forward something you really believe in. So I'm going to talk about the techniques we've been using to do quality control using genomics technologies in a development of a cell therapy for Parkinson's disease. So our mission is to develop therapies for incurable neurological diseases. I'm going to tell you about Parkinson's disease, but we're also working on multiple sclerosis and Fragile X syndrome, which is we're using as a model for autism. Our highest priority in all of these projects is to characterize the cells. Excuse me. We want to know what is in the culture dish. We want to know everything we can about the cells at every single stage of development. And that's not really trivial. We've had to develop a lot of assays ourselves, and we have a pretty high bar. So we want to know whether they're the right cell type. That is, we don't want to put a cell that won't work into a person. And we want to find out whether they're normal, whether they're going to have any opportunities to become cancerous, for example. Because any one of those things, if it goes wrong, is going to be devastating, not just to my project, but to the entire field. Okay, so I'm sure you all know this, but there are a few of you who haven't been involved in this as long as others of us. The cells I use are pluripotent stem cells, and they originally were derived from embryos in 1998. These are just 5 days after in vitro fertilization, using methods that had been developed in the 80s for mouse cells, so mouse pluripotent stem cells, which have been enormously helpful for making animal models and understanding development. But in 2007, Shinya Yamanaka published a technique for using another method, which is called reprogramming, in which you could use any cell type, essentially, a skin biopsy from a volunteer, growing up dermal fibroblasts, and then reprogramming them into pluripotent stem cells. And a lot of the work we've done has been in demonstrating, I hope without a shadow of a doubt now, that iPS cells, induced pluripotent stem cells, are in fact the same as embryonic stem cells, as remarkable as that may seem. Okay, so when you are working with pluripotent stem cells, there are enormous opportunities, and this tree represents a few of the things that these cells can become. So that means that you have cells in a dish that can become any cell type in the body. And the hard part is to make them become the cell type that you want for a particular therapy. So in this case, we're developing dopamine neurons in order to do a cell replacement therapy for Parkinson's disease. So the idea is to replace cells that die during Parkinson's disease. I'm not worried about the cause of Parkinson's disease. I'm not worried about slowing down the disease. We focus only on trying to reverse the disease. So the strategy, this is the substantia nigra right here. It's very small. Its target is up here. This is a striatum, a couple of structures that are known collectively as a striatum. So we make induced pluripotent cells from individual patients. We generate from those dopamine neurons, and then we transplant them back into the patients. That's what our plan is. It's really very, very simple. And we're basing this on historical success using fetal tissue. So in the mid-1990s and the early 2000s, the NIH funded 2 double-blind trials using fetal cells. Now, the fetuses that people used for these studies are really small. The human fetus that is used for this kind of work is about 6 weeks to 10 weeks old. And they physically dissected out a tiny bit of the brain, right here in the ventral part of this developing brain, that included the part of the brain that was going to become the substantia nigra. So I wanted to point out that I actually have deliberately shown that when you cut a piece of tissue like this out, you don't just have one cell type. You have all the stuff that's going on in that brain right now, including blood cells and things like, I mean, it's actually quite interesting. It's very complex at that stage. Nevertheless, when they put these cells into the target region, the substantia nigra, or the striatum, in some cases, these patients recovered and never required drug treatment again. So that's the remarkable part of it. The problem was that in a number of cases, too many in everybody's estimation, there was a dyskinesia that occurred, which is pretty frightening because it makes people sicker than they were before. It gives them another problem that is worse than Parkinson's disease and harder to treat. And so that's really why this closed down. This therapy has recently been rejuvenated by a group in Europe. They've started clinical trials doing the same thing, essentially, trying to do better quality control than was done in the past. But they are using fetal tissue, so they're trying to reproduce more reliably this work. It's a group called TransEuro. This demonstrates, just want to show you that there's, since these were done so long ago, and the patients have aged and died, it's been possible to look at their brains and find out whether those cells were still there. So in the patients in which they were, in fact, essentially cured of the disease, the fetal tissue, the fetal dopamine neurons are still there, at least 14 years in this case, but up to 20 years after the transplant. And one of the interesting characteristics, which is just shown in this little bit here, is that they continue to improve with time. So even over a very long period of time, these people, instead of getting worse, get better. So when it works, it's really remarkable. So here's the problems, though, the things we learned from these fetal tissue transplants. The main issue is quality control issues. It's very hard. You need three fetuses per side for a transplant. And it's extremely difficult. These embryos are very fragile, and you usually, even if you have the best of conditions for an abortion, the embryos are pretty chewed up by the time you get a hold of them. It's very hard to get the right piece of the embryo. They're not intact. So they have to use multiple fetuses, and the fetal tissue viability and in fact, the exact area of the brain that you can dissect is quite different for each fetus. So each patient is not going to get the same cells as the next patient. And then there was the unpredicted adverse outcome, dyskinesias, which has struck fear into everyone's hearts in this field. And of course, it's kind of difficult, especially in the US, to do any therapy using fetal tissues. These aren't fetal cell lines, these are fetal tissues, so they have to be harvested from the fetuses right before you do the transplantation. Luckily, there has been some information about that supports the idea that contaminants were responsible for the dyskinesias. And so that means that now that we plan to do cell therapy instead of fetal therapy, we can deal with those issues of quality control. We can know what cells we have in the culture dish and know what cells we're putting into patients. So the advantages of making dopamine neurons from iPS cells is that you can analyze them a lot before you put them into the patient. You can put the right number of cells into each patient. So you can put the same number of cells into each patient. We're not going to include any of the neuron types that cause dyskinesia. That's one of our quality control measures. There's no ethical issues with using patients' own cells. And I think probably one of the more important things is that there's no requirement for immunosuppression, because these are individually made from each patient, and they go back into that patient. So in a lot of ways, this is analogous to the CAR T therapy, in which you use a patient's own cells, with all the problems and the advantages. Okay, so our idea was to get a cohort of people. We have 10 Parkinson's disease patients. This is pictures of 8 of them here in this slide. We've put a lot of effort into differentiating the cells reproducibly from each cell line into the same population of dopaminergic neurons. We've had our first FDA meeting, a pre-IND meeting in February. We're planning to have our pre-IND meeting early next year, and it's a little hard to tell what's going to happen after that. But we're doing everything we can to make this as efficient as possible and hope to be in the clinic by 2018. Okay, so now I'm going to show you a series of slides in which we've tested every aspect of the process, and we've developed quality control assays that are not required by the FDA, and keep that in mind. This goes way beyond anything that the FDA requires. And I think this is really important because we also are interacting with the FDA, and we're hoping that we can convince the FDA that they require more stringent quality control than they currently require. Wish me luck. Okay, so I've identified 4 parts of this process. The first one is when you reprogram the cells, then you need to expand them in order to make enough cells so you can differentiate them. We don't need a ton of cells. We only need about 1 million cells, so we're not talking about bioreactors, we're talking about 6-well dishes. So that's a real advantage of Parkinson's disease. We don't require a lot of scale-up. It's also the advantage of autologous therapy, because each patient, we only need X number of cells, just that one time, or maybe 2 times. Okay, so let me tell you what we've done about each one of these. And this isn't in chronological order, but we've been sort of filling in the blanks as we go. Okay, so let's look at reprogramming first. There's been a lot of discussion about how reprogramming might introduce cancerous mutations. There's been a lot of papers published on how a particular reprogramming technique is safer than others. And so we decided we would just ask whether it made a difference how you reprogram the cells, and whether reprogramming the cells would cause any cancerous or cancer-associated mutations. And the answer is no, it doesn't. We did a huge study. Here we go into big data. We did whole genome sequencing of replicates of 3 different reprogramming methods. We used the classical method, which is to insert viruses using lentivirus, retrovirus. We used Sendai virus, which is a product from Thermo Fisher and does not insert in the genome. And we used messenger RNA, which is a transient expression of the factors. And this paper just came out at the end of last year. Actually, it came out this year, sorry, February this year. And it is data-rich, and the conclusions are unmistakable. That is that we did not find cancer-causing mutations. And I deliberately collaborated with a colleague from the Craig Venter Institute, because they know more than anybody else, at least in my environment, about what mutations are of concern for cancer. So we got a clean slate. We are not certain, we're going to keep checking, but we know that we can now focus some of our concerns in other directions, in other stages of the development. Okay, so the next issue of quality control is to know that you have pluripotent cells that are normal in a culture dish after you've reprogrammed them. In other words, we're now looking at sort of functional assays for them being pluripotent. There's no point in continuing if the cells aren't going to be able to develop into the cell type you want. So the first thing, and I think this has probably been the highest impact of our work in this area, has been to develop a bioinformatic assay for pluripotency based on gene expression profiling. We call it Pluritest, and it's a diagnostic test for pluripotency, and it has worked out simply because it's simple. We wanted to have the equivalent of an iPhone app for pluripotency for cells, and this is the closest we could get. Essentially, you create a gene expression profile, you upload the data without any analysis to our website, and you get a report which includes a diagram like this, which you can put straight into your publication. That's what people do. The idea was we started with 400 pluripotent stem cell lines that had been tested by the other method, which is a teratoma assay, which uses a mouse and shows that the cells can differentiate into all 3 germ layers. And we generated gene expression profiles for all of those, and we developed a model. And now what we're doing in this algorithm is looking at how similar those cells, these queried cells, are to our model. And that's where they map here. There's a pluripotency score, which goes up here, and there's a novelty score, which accounts for other cell types, but also you can end up with cells that are pluripotent but abnormal. So this has proved to be extremely popular. A lot of mice thank us for this. We think we've saved something like 14,000 mice this way. We now have 776 registered user sites in 29 countries. And as I said, we have 14,500 around now data set uploads, and they continue to be used almost every day. Somebody uploads a data set. We licensed it to Coriell in January so that we could step back and do some other things because we've maintained this ourselves. That we don't know yet how long we're going to be doing that license. We kind of liked controlling it ourselves, so we may end up doing that again. Or we may end up licensing it to somebody else. The point is it's been really effective and very popular, and it has saved a lot of work for a lot of people working in this field. Okay, the other issue about iPS cells is whether they have their epigenomes are correct or not. And I'm going to tell you right away that the answer is no, but we don't know whether it matters. So we did another very large study on we always use hundreds of cell lines if we can. Our sequencing paper, just because it would cost too much, we had to use tens of cell lines instead of hundreds. But generally, as you see from the author list, we get cells from everybody we can possibly. We want to know what the breadth of variation is among pluripotent stem cells from all over the world. So what we found, we used a DNA methylation assay that's based on an Illumina chip, and we could now look at 450,000 different sites of the genome and ask their methylation status. So what we found was that there were only a couple of things that were different about iPS cells from somatic cells. And they might be important, but so far we can't figure out how they're important. One is if you're reprogramming female cells, there's a high probability, you have one active X chromosome because there's X inactivation in us, in every somatic cell. When you reprogram them, the second X chromosome almost always becomes active. Now, we don't know whether that's going to affect therapy or not. It doesn't affect differentiation. It doesn't affect any of our downstream assays. The other thing that happens is imprinting is lost. Now, imprinting is the expression of certain genes by either the maternal or the paternal, by the alleles. And that just goes out the window. So we don't know whether that remains important for differentiated cells either. So those are things we're keeping in mind as we go forward. But for now, we don't see any reason why. The main point is that we check your cells. That's the point. Characterize them. Okay, so we have the Pluri test assay. I'm going to give iPS cells a green light here because we have the Pluri test assay, and it's very easy to do DNA methylation profiles. So if anything untoward happens, we will know at this stage later on if it affects the outcome. Okay, so here's the expansion phase. Now this turns out to be the most dangerous part. And the reason is that when you have cells growing in a culture dish, there is selection for cells that grow better or that resist getting killed. So in that sense, it's very much like evolution in the Galapagos Islands. And I think about my incubator with all its dishes in it as being the Galapagos Islands. Something different is happening in each one of those dishes. They're evolving differently. So we asked whether iPS cells are epigenetically stable. This refers back to the paper I just talked about. The answer is not really. They do change in the ways I told you about. So are they genomically stable? This is the paper we published a few years ago doing a copy number variation analysis on, again, hundreds of cell lines as they grew in culture. And we discovered that there were some things that would go awry. We found that there would be duplications of particular genes over and over and over again. Those duplications are acquired with time. The cells start out okay. One culture dish might end up with one duplication. Another culture dish of the same cells could end up with a different duplication. Okay? The Galapagos Islands. We also found something that was kind of alarming because we were working on methods developing methods so we could enhance the stability of the cells in culture over long term. And we discovered if we grew the cells for a really long time, like over a year or so, but it's in some ways this is equivalent to what other people are doing with these cells, we started seeing mutations that are clearly associated with cancer. In this case, it was deletion of p53. Which, as I'm sure you all know, is a common characteristic of virtually all cancers. So things can go very wrong. And so you have to monitor the cells often in order to make sure that they're not going awry. Okay, so the answer is they're not necessarily genomically stable. There are selective pressures in different dishes that leads to fixation of mutations, and in fact, we sometimes get mutations of p53, which is alarming. So I've given expansion a sort of half green, half red indication to show that this is an active area in which we have to monitor the cells closely. Okay. So what are we doing about differentiation? So this is now into the part in which we want to be compliant with the FDA, but we also want to have a higher bar than they require. So what we've done, this is our plan. We start with the iPS cells, and we do these release criteria. We have to show that the cells have a normal gene expression profile, that their epigenome is acceptable within limits. And we also do whole genome sequencing on them to make sure that, because there's no other way, really, to detect things that might be bad. It turns out none of these things are really hard. They just require like a very large terabyte server and people who know how to do bioinformatics. No problem. So that's one checkpoint. The second checkpoint is a median point at which there is an early stage of neural induction called floor plate induction. After 11 days after we start the differentiation protocol. We do the same things again. And we're developing a Pluri test-like test for this intermediate stage. So the Pluri test is based on looking at hundreds of samples. So we're acquiring the samples that we know are, by other criteria, in this case it's that they differentiate correctly, are the cells we want. And then we're going to create an easy assay so that we can check the cells quickly. We also can freeze the cells at any of these stages, which is really important because we need to put them away while we analyze them. The stage at which we transplant the cells is 25 days, and they are immature dopaminergic neurons. The reason we use immature cells is because if you try to dissociate the cells in order to transplant them, you break off their axons, you have enormous cell death. So we want to pick them at just the right stage. We want them to be just about done with their differentiation, but not quite. They haven't quite gotten these elaborate processes that they form in culture. So we're developing another test, which we call the dopamine neurotest, which is based on RNA sequencing or another assay for gene expression, whole genome expression. And then we do the epigenome and the genome again. Now this is really important because these are the cells we're going to put into people. So we want to make sure that the cells pass all these tests, as well as the things I haven't mentioned, like that the FDA requires, like FACS analysis. We not only do whole genome sequencing, but we use another method, SNP genotyping, to get a quicker idea of what's going on in the cultures. And then we take the cells further beyond the transplant stage until they're more than 60 days old, and then we can use electrophysiology to confirm that they're the right kind of dopamine neurons. So this profile here, shown in the electrophysiology this little diagram here, is typical of this particular type of dopamine neurons. And this is based on slice cultures from mouse. So as far as we know, this is a signature of this particular type of dopamine neuron. There's lots of dopamine neurons. We need to have the right one. So we're also working at this stage on single-cell biomarkers. We want to find out, and it's actually kind of easy because when you do a patch clamp on a cell, you just suck it up into your pipette, and then you do RNA sequencing on what you get. These are the two people who are involved in leading this project. This is Andreas Bratt-Leal. He's the director of the Parkinson's Project. And this is Roy Williams, who's our bioinformatics expert. Roy's been working with me for, I think it's 11 years now. So we've sort of grown up together in this space. Okay, so to summarize, ensuring safety is the highest priority. And the most important thing is to characterize the cells. And this is me on my soapbox, yelling at everybody whenever they give a talk, talking to the FDA, talking to whoever it is will listen to me. You have to know what's in your dish, and you have to use, there's no assay that you should just skip because you can't do it or you don't understand how to do it. You need to find out. Okay, so I just want to turn in a very minor way to the issue of funding. We've had a lot of funding from CIRM. This is a list of the grants that we've used to develop the gene expression profiling, the SNP analysis. You notice that a lot of them are defining pluripotent stem cells, ensuring the safety of cell therapy, developing new assays, and doing sequence analysis. And CIRM has funded all of this. But the most important funder of all this research, and this is unique in I think everyone's experience, is the Summit for Stem Cell Foundation. Let me tell you a little bit about them. So they started in, this is a nonprofit foundation that was started in 2010 when I met with the head of the Movement Disorders Clinic at Scripps Clinic, which is across the street from us. We're not affiliated, but they're both called Scripps. So they came over and asked me whether there might be a stem cell-based therapy for Parkinson's disease for her patients. And I said yes, because it wasn't that difficult to think about this. It's pretty straightforward, it's simple. The details, of course, are a little harder. But we didn't have any funding, so I said the problem is the funding, because I'm not going to get NIH money for this, and even CIRM is not going to fund something that's this speculative at this stage. So in 2011, Sherry Gould, who's a nurse practitioner, took 15 people up to the top of Kilimanjaro, and 3 of them had Parkinson's disease. So by doing that, they raised $300,000, which was the amount we decided we needed to initiate the project. That's why it's called Summit for Stem Cell, in case you're wondering. So we've had lots of fundraisers since then, and when I say we, I really mean them. It's been an amazing group. They sponsored concerts. There's been private dinners at chefs' houses, asking people for money, social media. They climbed to the base camp of Mount Everest. Again, there's a movie about this. It's pretty cool. You can see people with noticeable tremors walking up these rocky paths. There are people with Parkinson's disease who made it up to the base camp of Mount Everest. And they're planning this fall to trek to Machu Picchu in Peru. Over the last 5 years, they've raised more than $3 million, and that has allowed us to get to where we are now. There are thousands of donors worldwide. We have donors from Australia and Europe and Asia, as well as the US. They've done a remarkable job, and they continue to work on this. Meanwhile, we've finally received our first CIRM grant to support this project, because we had finally had enough preliminary data to convince them that we knew what we were doing. But we couldn't have done that without Summit. And I just want to close by showing the people again. These are the patients that we've derived iPS cells from. These are the people that we're planning to treat. And having the patients involved has been a really remarkable experience. They come to our lab. I think Dane's probably met them, my intern. They come to my lab, they're involved in the fundraising, they understand the science. It isn't beyond them at all. They have no problem explaining it to other people. And that's made a huge change in the way that it's not only allowed us to raise money, but it's also allowed us to educate people about what a real stem cell therapy looks like. And that's important because there was a recent report that the number of unregulated stem cell clinics, that is people who use fat cells, for example, or bone marrow mesenchymal stem cells to treat autism or Parkinson's disease, has risen to 570 in the US alone. There's no FDA regulation of this at all. So it's very important for us to also educate people about what kinds of stem cell therapies work and what kinds of stem cell therapies are unlikely to work and might even be dangerous. Okay, thank you.